Okay, this is our last video regarding cell communication or cell signaling, and it is regarding the cellular response. And so after watching this video, after reading your text, I want you to be able to describe the difference between nuclear response and cytoplasmic response, describe the function of a transcription factor, list several ways in which a single cell, there should be the word cell there, might cause a single signal might cause different responses in different cells. List the steps involved in epinephrine signaling and describe how the signal is amplified. And describe how the cholera toxin is an example of why it is important that signaling pathways can also be turned off as well as be turned on. Here is our broad overview of the signaling pathways here, where we have a signal is received by a receptor, and then we have that signal transduction pathway leading to a cellular response. And so we are focusing here on cellular response. And cellular responses are extremely diverse. Anything a cell can do is pretty much a cellular response. But we can break it down into two broad categories. The first is a nuclear response. Basically what that means is we're turning on genes. So here we have an example. We have a growth factor binding to a receptor. That activates a phosphorylation cascade that then goes on to activate a special type of protein we haven't talked about before, a protein called a transcription factor. Now, transcription factors are proteins that are capable of binding DNA at very specific places and activating the transcription of a specific gene, leading to the production of its mRNA, which will then be translated into a protein. So what we have here is an example of a signal that leads to the activation of a gene within the nucleus, and then eventually the production of the protein that is encoded by that gene. The other example is a cytoplasmic response, where the protein already exists within the cell, within the cytoplasm. So here it is, but it's not turned on. So a cytoplasmic response is, here's the signal, receptor, we have our signal transduction pathway, and then that leads to the activation, the turning on of a particular enzyme that then does something. Now, maybe it breaks down a molecule like glycogen, so maybe it's some sort of enzyme that does that. Maybe it alters the cytoskeleton so that you have something maybe like cytoplasmic streaming or something like amoeboid movement. There's all sorts of things that, all sorts of ways that a cell can respond to a particular signal. Here is a comprehensive example, and this comes all the way back to that first slide in that first video regarding the impala running away from the cheetah. In order for its muscle cells to have enough energy in order to fuel the amount of contraction that's required for that impala to just really escape the clutches of the cheetah, is that it needs a lot of glucose. And to get that glucose, it needs to break down molecules of glycogen, right? Glycogen is a bunch of glucoses stuck together, and there's an enzyme called glycogen phosphorylase that's responsible for breaking down glycogen into glucose molecules, but normally it's turned off. So the pathway that leads to its activation, to turn it on, to break down glycogen into glucose molecules, is this pathway here, where we have a specific G protein coupled receptor that receives that epinephrine signal, that then activates the G protein. That G protein activates an adenylyl cyclase. That adenylyl cyclase makes cyclic AMP from ATP. The cyclic AMP activates protein kinase A. Protein kinase A phosphorylates and activates phosphorylase kinase. Phosphorylase kinase phosphorylates and activates glycogen phosphorylase, and therefore we break down a bunch of glycogen into glucose, and now there's suddenly plenty of fuel for that impala to use in its muscle cells to contract and run. This is also a fantastic example of the idea of amplification. Here we have one molecule of signal binding to the receptor that activates 10 squared molecules of G protein. Okay? That then leads to the production of 10 to the fourth molecules of cyclic AMP. Eventually that leads to the activation of 10 to the fifth phosphorylase kinases, which then activates 10 to the sixth molecules of glycogen phosphorylase, which leads to the production of 10 to the eighth molecules of glucose. Which means in the end, what we have is a single signaling event leading to a robust cellular response of the production of 10 to the eighth glucose molecules. Well, specifically glucose 1-phosphate molecules. So you will be responsible for knowing this complete pathway, although you don't need to know these numbers involved here. Now, different cells can respond to the same signal in different ways. There's all sorts of possibilities here. Here we have our baseline situation where we've got a particular cell, it has a receptor, it receives the signal, we have certain relay molecules that respond in a certain way. Fantastic. Maybe we have a different cell. Now that different cell, when it receives, it's got the same receptor, it receives that signal, but now instead of turning on this particular relay molecule, it turns on a different one. And then that relay molecule then turns on different molecules, and so leading to a second and a third response. Or we have crosstalk, and I mentioned something a little bit about this before. Another reason why we have these big, long, indirect signal transduction pathways like we see here is because it allows for regulation, or in this case, crosstalk, where what we have is, okay, we get that signal, it binds to the receptor, it turns on a signal transduction pathway, but this particular cell has another receptor that receives that. When it receives this signal, it sort of overrides this signal here, and it can then modify what that response might be. So that's response number 4. And then in even a 1, 2, 3, 4th different type of cell, there might be a completely different type of receptor that is on that cell's membrane that receives the exact same signal, but because it's a different receptor, it turns on a different signal transduction pathway, leading to yet a 5th different response. So there's an incredible diversity of responses depending upon the cell type, and it can happen in all sorts of different ways, whether we're turning on different signal transduction pathways or whether we have different receptors binding to that same signal. Well, next, now we've been talking about turning on particular signal transduction pathways, but it's absolutely essential that we can turn these signals off as well. And one example is cholera. So here is the normal situation in our gut. We have this particular pathway, signal transduction pathway, and a G dependent upon a G protein coupled receptor. And the cellular response is it leads to the secretion of salts into the intestinal lumen. And this is something that is normally important to occur. The problem is that if you are infected by cholera, the cholera produces a toxin, and that toxin, it interacts with the G protein and it inhibits the G protein's GTPase activity such that the G protein can no longer hydrolyze that GTP, can't turn it into GDP, which means that this G protein can't turn itself off. So in the presence of this cholera toxin, this G protein is constantly on, constantly on, constantly on, meaning regardless of how much signal is or is not here, this pathway is happening. It does not care what's happening to this receptor here. This G protein is constantly on, constantly turning adenyl L cyclase on, constantly making cyclic AMP, constantly activating protein A kinase A, and constantly secreting salt into the intestinal lumen. And the problem with that is you're creating a hypertonic solution within the intestinal lumen. All the water then floods out of your blood cells, floods into the intestinal lumen, and you now have uncontrolled diarrhea, which is extremely dangerous because you will potentially die from dehydration. Now, here is a bunch of ways that signal can be terminated. The first, the receptors bind to the signals bind to receptors reversibly, meaning once the concentration of receptors go down, you will have less and less receptors bound. Once the concentration of signals goes down, you will have less and less signal bound to receptors. Without the signal, then, the receptors revert to their unbound conformation. G proteins will catalyze hydrolysis of their own GTP, right? They'll turn themselves off by converting that GTP to GDP. All of the various kinases in a phosphorylation cascade will be dephosphorylated and thus turned off by phosphatases. And finally, phosphodiesterase will convert cyclic AMP to AMP. So basically, we'll get rid of that cyclic AMP. And so these are all the ways that these signals, once activated, can become deactivated. So any pathway that is started can also be ended. And it's just important to be able to end a pathway than it is to start it. Now, one thing that you are going to learn about molecular biology and biochemistry, it's all about the enzymes. And we have talked about 1, 2, 3, 4, 5, 6 enzymes in this one topic. And we're going to keep learning about enzymes. And so I want you to get in the habit of making tables. Anytime you learn about a new enzyme, bam, pop it into a table. Figure out what its basic function is, and then give me examples within the context of the cell. So I'll leave this table here for you as an example to go through. I will not fill it out for you. So hopefully by the end of this video, after reading your textbook, you can describe the difference between nuclear responses and cytoplasmic responses. You can describe the function of a transcription factor. You can list several ways in which a single signal might cause different responses in different cells. You can list the steps involved in epinephrine signaling and describe how the signal is amplified. And finally, describe how the cholera toxin is an example of why it is important that signaling pathways can be turned off.